Welcome everyone to our Tuesday webinar as part of our Phenology Week webinar series. We're really glad to be celebrating Phenology Week with you all. Phenology Week is simply a celebration of all things phenology with special webinars, observation challenges, and highlights of our observers and partners. This is the second webinar in our series this week, and we will be having a bunch more webinars later this week, and I'll run through what those are and where you can sign up for them at the end of the webinar. My name is Erin Postumus. I'm the Partnerships and Projects Manager here at the USA National Phenology Network at the University of Arizona. I'm really glad to be joined today by three researchers who are affiliated with our Nature's Notebook campaigns. We have Kim Pegram, who is the Director of Pollinator Conservation at the Desert Botanical Gardens up in Phoenix in Arizona here. Really glad to have Kim on the call today. Kim has been working with us on our Desert Refuge campaign since I think 2022 when we first started that campaign. We also have Nick Diechler with us. He is a research support specialist and PhD candidate at Cornell University, and he has been researching the hemlock woolly adelgid, which is part of our pest control campaign. And then we have Andrew Hipp, who is the herbarium director and senior scientist in plant systematics at the Morton Arboretum. And he has been working with us on the Quercus Quest campaign since we started that campaign in about 2022 as well. So really excited to have them with us. I did just want to start off by giving you just a brief orientation to our Zoom here today. So we are recording this webinar and we'll be sharing out the recording with everyone in a follow-up email. I encourage you, if you'd like to put in the chat where you're calling in from. We always love to hear about where you're located. You can feel free to put in, if you're already participating in one of our campaigns, maybe you're part of one of the campaigns featured here today, we'd love to hear that as well. If you have any questions for our presenters, we'd love for you to put those questions in the Q&A, and you'll find that at the bottom of your Zoom screen as well. And that'll just help us keep track a little bit better of those questions and make sure we answer them. But feel free right now to put in the chat where you're calling in from and if you're part of one of our campaigns or anything else you want to tell us phenology-wise. And feel free to please post on social media this week about Phenology Week if you want to share anything related to what you're seeing out there phenology-wise. It might be a little cold depending on what part of the country you're in, but we'd love to hear about what you're seeing this week. I did just want to start out too by just giving a little bit of an overview about what campaigns are. So if you're a Nature's Notebook observer, you can observe a whole suite of different plant and animal species. And we love for you to observe any plant or animal that's of interest to you. But we also have a number of kind of special collections of species that are what we call our Nature's Notebook campaigns. These are usually species that are of special interest to researchers or natural resource managers, and we really like to try to encourage people to collect data on these species. So if you collect data on one of those species, you can know for sure that your data are going to be used by these researchers and natural resource managers. And even though, any of the data that you collect really can be used by researchers, these ones we know immediately are going to have use. So it's a really nice way to make sure that your data are going somewhere and are going to be used immediately. And another part of campaigns that's really great is we will send you messages. So as long as you participate in tracking one of the species that are part of the campaign, you're automatically counted as part of that campaign. But you can also opt into special messaging, and I'll show you how you can do that at the end of the webinar as well. So with that, I'm going to turn it over to our first speaker, which is Kim Pegram. She's going to talk about Desert Refuge.
Okay, I'm just gonna get us going on the screen share. Are we good to go on that end? Okay, great. Thank you, Erin. If I don't, remember to say it. Erin and the rest of the team at NPN have been so critical, so important to our campaign and the data that we've been able to collect, and we really appreciate their support and also the support of all of our volunteers. So in case I forget to say it, I just wanted to open that up. So what I'm going to talk about today is the campaign Desert Refuge that we've been working with NPN. Um, a little bit of a background, what led up to it, and what important questions we're trying to answer, and then talk a little bit about the campaign and what we found out so far. So, jumping into it, my job at Desert Botanical Garden, or part of my job, is running um, a pollinator, a monarch and pollinator conservation initiative we call Great Milkweed Grow Out. It has three components. One is outreach and education, just like this talk I'm giving right now. This is part of outreach for the program. We also grow a lot of pollinator plants, plants that are beneficial for pollinators. We've noticed a real lack of availability of pesticide-free plants, so we grow a lot, thousands of plants every year that get distributed mostly in the Phoenix area. And then we do research. We have research plots at the garden, but we also do community science, just like this campaign, all with the goal of helping us better understand pollinators in general, pollinators of the Sonoran Desert, and how they interact with plants. So there's, hundreds, thousands of species that I could work on pollinator wise. And I get asked sometimes why monarchs? Monarchs, everybody knows them. And maybe that seems a little boring, but monarchs, because everybody knows them, are kind of a gateway to gathering interest from the public, from people. That do community science, that use Nature's Notebook. And so I find that working with monarchs does have it allows us to capture the imagination of more people and to draw more people in, and captures a lot of public attention. One of the cool things about monarchs, honeybees is the other one that you'll hear a lot about, is that we can get a repeatable population measure every year because most of them overwinter in Mexico and California through after the migration. Um, the we are able to get a population count, an estimate every year. And then the things we do for monarchs, planting milkweed, planting other pollinator plants, reducing pesticide use, all of that is helping a whole variety of pollinator species. So that's one of the reasons we do work on monarchs. So most people know that monarchs undergo a pretty phenomenal migration. There's roughly two flyways in the US. We have the eastern population mostly lives east of the Rocky Mountains in the spring and summer, and then they spend the winter in Mexico. That's the much larger population. And then we have a smaller population in the west that overwint or that lives west of the Rocky Mountains roughly, and then spends the winter in these smaller sites along the coast of California. But one of the interesting things about Arizona is that we have a little bit of both. We are kind of in between the two flyways. And this is actually data from another community science project, Southwest Monarch Study. Sorry, I'm going to promote another one, Erin. Southwest Monarch Study that allows individuals to tag monarchs with like a sticker-like tag, doesn't harm them. But monarchs tagged in Arizona have ended up in both Mexico and California. And so we are right in between these populations. Um, But the other interesting thing is that we also see monarchs hang around in Arizona for the winter. Not very many, but a small population, enough that has us going, what's up with that? And, that's how science is born. So I'll now talk a little bit more about that overwintering population in Arizona. Oh, yeah. So there's a picture of a tag on a monarch. So when monarchs are in these overwintering sites, the larger sites in Mexico and California, they are in something we call reproductive diapause. That means they're not reproducing. They're in this hormonal state where their focus is solely on surviving the winter. So they first migrating south or west, and then this entire butterfly lives the whole winter not reproducing until the spring comes. And when the spring comes, that's when they move out of this. They have this hormonal shift back to reproductive status, and they're more active, they're feeding, and they're seeking places to lay eggs. So this is what's happening in a normal overwintering site. So we have this small overwintering population in Arizona, and we would expect the same thing, right? Well, that's not exactly what we're seeing. Um, we see some, or anecdotally, before we started this project, we saw them being pretty active. We see a lot of adults flying around. We'd see some mating going on. That's what's happening in the first picture there with the two monarchs. We'd see them basking in the sun, which isn't unheard of in the overwintering populations, especially as it starts to warm up. But they were feeding, they were flying around, and then we would find caterpillars in parts of the winter, which was telling us they weren't going through the same reproductive diapause. And there was some evidence they were not reproducing for part of the winter, but it was mostly all anecdotal and things that we had seen from our time observing monarchs in the garden and hearing reports from other people. And so what we really wanted to do was get a handle on what's going on with our overwintering monarchs in Arizona. What are they doing? Are they reproducing? Are they active? Are they visiting milkweed? Milkweed is the host plant for monarch butterflies. It's the only thing they lay their eggs on and their caterpillars eat. And so one of the ideas for why we have an overwintering population in the southern parts of Arizona, central and southern parts of Arizona, is that because our milkweed doesn't die back in the winter like it does in other places. Um, so this was one of the main reasons why we really wanted to get this campaign started with NPN. So some of the key questions when we created this campaign was, first of all, when? When are the monarchs? When are we seeing them in central and southern Arizona? We knew northern Arizona was too cold. They weren't really spending the winter there. So the focus so far has been mostly the Phoenix and Tucson areas. We'd love to have people that are more rural, that are more outside the city areas, but it's community science. It's where the people are. We wanted to know, when are they breeding? When might they be mating? When might they be laying eggs? And also, if this is tied to the milkweeds not dying back, what is the phenology of milkweeds in Arizona? What is the timing? When do they have leaves? When are they blooming? Maybe that varies between milkweed species. These are all things we wanted to determine with the campaign. Um, and so we've asked participants using Nature's Notebook to track milkweeds that are in their backyards. So we, over the years, have encouraged a lot of people to plant milkweed in their yards, and a lot of people have. And so we encourage people to choose a milkweed that is readily accessible to encourage continued data collection. And then roughly weekly to record the phenology of your milkweed plant or multiple milkweed plants if you have them set up that way. Any caterpillars seen, and that would be a measure of reproduction for the monarchs. If they were seeing caterpillars, then they have reproduced lately. And then any adult monarchs you might see flying around. And that was the kind of simple ask of those people in central and southern Arizona. So I'm going to show you some of the data. We started a pilot study in 21, 22 with a limited number of people. And now I'm really glad that we did because it seems so far that 2021, 22, that winter was the last winter we saw a break in breeding that we had seen anecdotally prior to that. So what I'm showing you here is a graph of activity that is seen, or the presence of caterpillars that have been seen by our Nature's Notebook observers. So each dot is a time that an observer has seen a caterpillar on one of their milkweed plants. And so so we're part of the way or almost through the fourth winter. Um, so you can see that data really only goes up through March anyway. And so in 2021 and 22, we saw the break that we expected. When it was really cold, we expected that the monarchs weren't going to be reproducing, that we weren't going to see very many caterpillars, and we saw that break. And then in 2022 and 2023, we didn't. They kept reproducing the entire winter. Same thing in 23, 24, and 24, 25. It seems to be that way too. Um, we're also seeing a later start every season. And that is very likely, and all of this is speculative. This is just, early data. Hypothesis is that it's stayed warmer in the fall here in Arizona. We're seeing warmer temperatures, and monarchs have a temperature threshold. They don't like the heat. Um, so we're seeing them come into these areas later and later, it seems. Um, so you might be looking at that big gap in the 2021, 2022 data and thinking, well, maybe people weren't just, maybe they just weren't finding caterpillars. Maybe they weren't checking their plants. It was a small pilot study. Well, so this is the amazing thing about Nature's Notebook, and that has allowed us to capture data that we otherwise couldn't do, is we get absence data. Absence data is when a person is recording, is out there with, Nature's Notebook checking the boxes, but doesn't find anything. This is what makes NPN and Nature's Notebook different from iNaturalist or any of these other community science or some of the other community science projects, is that we are able to get this absence data. And so we can see this clear, people were recording data all the way through the winter and still didn't see any caterpillars. Same thing with the early starts of the start of the season in these later years. We have people recording data the entire time, but not actually seeing caterpillars until late October. And so this is what really, I think, sets apart this campaign, and one of the the benefits that we've really seen from this collection, data collection, a community science project. I will just show you two. Oh, sorry, and I hope I did explain the and I maybe I didn't. The gray dots are the negative data, are the absence data where people were recording. Um, we're collecting data, but they didn't find anything. Um, we do see that the monarch adults are active throughout the winter, or we're at least seeing them. And so that's important too, that we're seeing adults, but not the reproduction in that first year. And in the same, we are seeing uh, seeing adults mostly active. They seem to maybe be ending late or sooner this year, but we'll see. Where this is why we need to keep collecting data. We encourage people roughly September through May to be collecting this data. So that's my quick wrap up of what we've been doing. Thank you to everybody, all of our volunteers that have been collecting data along the way, and for all of our funders, of course, and Aaron and Teresa at NPN. Thank you.
Thanks so much, Kim. That was wonderful. If you have questions for Kim, feel free to put those in the Q&A, and then we'll get to all the questions at the end of the presentation. And I will also put the link for Desert Refuge in the chat if you'd like to learn more about that campaign. Now we're going to be moving from Arizona up to New York, and so I'll pass it over to Nick.
Okay, let's see here. Find my presentation. Okay, how's that look? Okay. So I just want to start by thanking Aaron and all the folks at the Phenology Network for having me here today. Really excited to chat with everybody a bit about some of our work up in New York. So I'm going to be talking about the pest control program and how phenology informs pest management. So the organism that I work with is called the hemlock woolly adelgid. So this is for anybody that's not familiar with this, and it seems like there's a lot of folks across the country that aren't in hemlock territory. So this is a small aphid-like insect that feeds on Eastern and Carolina hemlock, which are both native to the East Coast. Eastern hemlock has a pretty wide distribution. Carolina hemlock has a small range in the southern Appalachian Mountains. But this insect feeds on hemlock and kills it. This is introduced from southern Japan to the East Coast. And again, this is part of the pest control program. So you can see here, it's pretty widespread throughout the East Coast. Was first introduced in the Richmond, Virginia area, and is spread throughout the Appalachian Mountains up into New York. It is getting into Maine, but is also in Nova Scotia and Ontario. So impacting hemlocks throughout that range. And the reason we really care about this pest is because hemlock is what we call a foundation species. So throughout that range where it exists, this tree is really important to a lot of different organisms. It creates this really unique habitat, this unique ecosystem that a lot of birds, arthropods, and different critters really depend on, keeping streams cool for cold water fish like trout and things like that. So really when we're trying to measure the impacts of this invasive pest or manage for it, we're really trying to understand how it's impacting all of these other species. And really, when we're trying to protect these habitats, we're not trying to just save the tree, we're trying to save a whole bunch of different species. So how phenology comes into play with our research is we work predominantly with classical biological control. So we work with three different predator species that are actually native to Western North America. There's a native lineage of hemlock woolly adelgid on Western hemlock from Northern California up into British Columbia and Southeast Alaska. And these three predator species are specialists on hemlock woolly adelgid. They feed on very specific developmental stages of the adelgid, and our work is trying to research, learn more about these insects, and move them to the East Coast to try to control hemlock woolly adelgid. So these are specialists, they only feed on hemlock woolly adelgid, and the goal is to try to establish populations of these so we can learn to live with the adelgid, manage it, and try to protect hemlock trees. So hemlock woolly adelgid, I'm not going to get too much into the nitty gritty, but just kind of to quickly touch on why understanding the phenology of this insect is really important for biocontrol and management. So hemlock woolly adelgid has two generations that occur on hemlock throughout the year. There's this overwintering generation. So this is a really weird insect. It actually has its dormancy in the summertime and then comes out of dormancy in the fall, grows throughout winter, laying its eggs in late winter, early spring. And then this gives rise to a second generation of the adelgid that has no dormancy, lays eggs, and then kind of starts that process all over again. So most of the action with this insect happens throughout the winter time. And the predators are also present during the winter, feeding on hemlock woolly adelgid during different life stages. So when it came to working with these predators on the East Coast, New York is a big state. Hemlock occurs throughout a huge variation of different climates on the East Coast. And being able to release these predators and then monitor for their establishment is really dependent on the phenology of the pest. So when my boss came to me at the time about 8-9 years ago and said, hey, you need to figure out how to monitor the development of this insect across the landscape, I was thinking, geez, I'm one person. This is a big area. So I thought, there's folks that are probably already doing this kind of work. And I knew from developing a phenology trail at a local preserve near me about 10 years ago that the Phenology Network existed. So I got connected with the New York Phenology Project, Carissa Battle, and then started working with the the National Phenology Network on developing protocols to look for these very specific points of hemlock woolly adelgid life cycle. So the key points that we are trying to identify is when hemlock woolly adelgid comes out of dormancy in fall, because that's when we want to start releasing beetles, but also monitoring for those beetles, their presence in the landscape to confirm establishment. And then also in the springtime, looking for egg laying to begin on the hemlock woolly adelgid so we can monitor for establishment of beetles, but also start releasing those silver flies. So this kind of turned into a project that was led by the National Phenology Network, where they developed these phenological forecast maps, which were aimed at predicting when these certain developmental stages of these different pest species were going to be occurring. So that way we could direct citizen scientists to get out there and look for these stages and confirm them. So this is a map that's actually showing emerald ash borer. This is from the paper. So based on growing degree day accumulation. So insects, their development is dependent on temperature. So we can measure insect development using something called growing degree day. And this kind of allows us to look across the landscape, how these degree days are accumulating. And we can direct folks to go out into the field and confirm whether or not they're seeing these different life stages based on the degree day accumulation. So these were created for hemlock woolly adelgid, and throughout the years of 2017 and 2018, I actually went out all over New York State, training folks how to observe for hemlock woolly adelgid, these different important phenological stages of hemlock woolly adelgid, and getting folks out there looking and contributing data to Nature's Notebook. And this allowed us to look at these phenoforecast maps and see how accurate they were. So a map was created for HWA, volunteers were trained, and we actually found that the predictions were accurate for egg laying for up to 87%. And for the active dispersal stage, which is timed with that egg laying in the spring, those maps were accurate 92% of the time. So this research allowed us to, one, create the maps, but also then use citizen science data to confirm that these maps were accurate so they could actually be deployed for years following. So after these maps were created, we continued to collect this data over the years. We really didn't have a lot of plans to publish this data. We weren't really sure how much we were going to learn, but we knew that this data was really, really important for just us implementing biological control. So we were using this year in and year out to plan our biological control releases, to plan our survey activities. But then we started to accumulate a lot of this data, and we started to notice a lot of different patterns that were really interesting to us, and started to answer some kind of key questions around hemlock woolly adelgid biology that folks had really been puzzling over for a long time. So we published this paper early last year, kind of summarizing a lot of this data that we collected over the years. So basically, this is an image of hemlock woolly adelgid in the top left-hand corner there in frame A. That is what a dormant hemlock woolly adelgid looks like. It's really tiny. It's about the size of a grain of pepper. And it just kind of looks like this little sesame seed with this white halo. But when it breaks dormancy and it begins to develop, it starts to plump up. It gets bigger, and you can actually start to see these signs of this red hemolymph showing through the sutures. So this is a really, really kind of specific phenology project for people that are really interested in looking at stuff through hand lenses and microscopes. But surprisingly, we got a lot of folks that were really interested in doing this. So basically what we were trying to do is look at the breakup in this dormancy and egg laying, and then this would allow us to release predators and also look for their establishment. So when we started to accumulate this data, we started to see some really interesting things. Some things that weren't overly surprising, but it was really nice to document them because there's a lot of publications out there that are these snapshots of areas where that do these in-depth phenology studies over a single year, and they kind of describe what the phenology looks like, but it doesn't really capture this year-to-year variation. And this was something that we saw throughout New York State. Based on where you were in the state and from year to year, we saw this really huge variation of when egg laying started. So that was really interesting to us. You know, things could vary quite a bit from year to year. And this is really, really important for releasing those predators, because if we release them too early and there's no food for them to eat, establishment's going to fail. They're not going to do well. But if we release them too late, they're not going to have enough food to complete development and become established. So we really rely on this data for releasing these predators to increase the chances of establishment for controlling this pest. But another thing that we noticed is that there was very little variation in the onset of fall development. And this is something that's puzzled researchers for years. People have used all different sorts of language and descriptions to describe this estivation break, but we were using a very specific definition. As soon as we were seeing development, we were calling that estivation break, as soon as those insects began feeding. And the fact that we were seeing this happen at almost the same time every single year with a little bit of variation pretty much was, it was starting right that last week of September. And this is really important because with lab rearing one of the predators, the Laricobius beetle, we get early emergence in the lab. And we really struggle with keeping these predators alive in the lab. And they're really, really valuable. They're expensive to rear, and we want to get them released onto the landscape as soon as possible. So by knowing the earliest point at when these insects are actually coming out of their dormancy, we can release them as soon as possible, or we can get food into the lab to feed them. So the fact that we were seeing this happen at the same time was intriguing. So we actually set up this comparative study between Virginia and New York, working with our colleagues at Virginia Tech in Blacksburg, Virginia. And we looked at development over a couple different sites between Virginia and New York. And we actually found that that this dormancy ended, these insects began developing on almost the exact same day between Virginia and New York. So this is something folks have been really curious about for a long time. We still don't really know the mechanism of what's triggering these insects to come out of dormancy. You know, this happens right around the equinox. It could be something with photoperiod. So this has sort of opened up a whole new set of questions for researchers to really begin thinking about when it comes to the development of this insect and how that speaks to biological control. And this is information we never would have been able to gather without working with citizen scientists contributing data through this program with the National Phenology Network. So just a quick couple points, take-home messages here for folks before we move on. We found that HW egg laying was variable between sites and within years and between years, and that annual data collection was really important and needed to track this egg laying for release of predators. We found that dormancy break is consistent across years and across the landscape, which was brand new to science. We had no idea that this was this consistent. And this phenology data collected through citizen science led to these breakthroughs in understanding basic pest insect biology. And this is still information that we're using every year to plan our biological control releases and our surveys. So we really rely on this data collected by folks to help improve our research and our release program. So lots of folks to thank. A lot of our funding comes from the New York State Department of Environmental Conservation and U.S. Forest Service. So really grateful for that. And then all the partnerships that have been formed around this project, but also a lot of those relationships with the volunteer observers. You know, we've been working with some of these folks for years, and it's been really an honor to work with a lot of people contributing this data. So that is all I have for now. So thank you so much.
Thank you, Nick. Really interesting results. And it's so cool that our observers can be part of a project that has such direct management applications. That's really great. So I'm going to put the pest control campaign link in the chat. And there are, in addition to the hemlock woolly adelgid, there are 13 other pests that you can observe as part of this campaign as well. All right, I'm going to share screen now with Andrew's slides. So next we are going to the Midwest, over to Illinois, and we're going to hear about Quercus Quest. Let me just share really quick.
Thank you very much, Erin, and thank you all for coming today. It's great to see everybody here and have a chance to talk with you a little bit about how National Phenology Network and your data are contributing to our project. First of all, I'd like to thank Erin and Teresa Crimmins, who've been fantastic to work with. This is part of a big international collaboration focused on understanding how oak biodiversity shapes diversity of the entire forest. So we're working with colleagues at a number of universities in the US and a number of universities in China to look at interbreeding white oaks across Eastern North America and also East Asia, and understand how that pattern of interbreeding and the evolutionary history of oaks shapes the symbionts that depend on oaks. These are a group of the PIs who are involved in the project. And on the next slide, you'll see a lot of the people who are doing the active work. Erin, if you can move, yeah. This is the graduate students, postdocs, undergraduates, research assistants who are doing a lot of the day-to-day work, and also writing the papers, analyzing the data, etc. Let's look at the next slide. So this project, Quercus Quest, is really part of this collaboration investigating how oaks shape the communities of the organisms that live on oaks. And so we are looking at the traits of a set of Eastern North American and East Asian white oaks, and then what fungi live in their leaves. Those are the endophytic fungi. What fungi live within their roots. Those are the endophytic fungi in the roots. What fungi live in symbiosis with the trees on their roots. Those are the mycorrhizal fungi. The gall wasps that form communities on the trees. The leaf herbivores that eat the trees. And then the other trees as well. All of them depending on oaks, and all of them relying on both the traits that have evolved on the oak tree of life and also the gene flow between species. At the next slide, we can see the distribution of the trees that we're working on. Now in Quercus Quest, we're focusing on all the Eastern North American oaks that live in sympatry with Quercus macrocarpa, which is bur oak. That's the upper left-hand map here. What you're seeing, these triangles, these are collecting sites where we have collected trees with genomic data across the range of bur oak, and with it all the co-occurring white oaks. So the most common of these being white oak itself, Eastern white oak, Quercus alba, swamp white oak, Quercus bicolor, post oak, Quercus stellata, and then chinkapin oak, Quercus muhlenbergii. In addition to that, we've gotten a handful of other white oaks. From each of these trees, we gather data, one set of data being the genomic data, which tells us which trees are actually hybridizing. And on the next slide, you can see an example of two of these species, the bur oak here on the left side and the white oak on the right side, that often hybridize. And when they hybridize, you find things that may be morphologically intermediate in that first generation. But in addition, they're ecologically intermediate. So those hybrids have the opportunity to move symbionts between species. They can actually act as a conduit between species. The next slide. As we look at the Eastern North American white oaks and the East Asian white oaks, there has been gene flow going on between these species for thousands, probably millions of years, but for sure we can document it over thousands, and in some cases even more ancient gene flow than that. Now, hybridization is often thought of as just a merging of species. It's really not in the oaks. What we see with hybridization is that genes that move between Quercus alba, Eastern white oak, and Quercus macrocarpa, bur oak, for example, in those F1 hybrids, then those hybrids have a chance to back across to one parent or the other. And as they do, little by little, you don't even realize that they're hybrids any longer. They look exactly like, after a few generations, the one parent to which they've back crossed more often. But that movement of genes then between species is part of what enables oaks to survive and adapt to climate change and to environmental gradients across the continents. When we look at what potential hybridization there is between species, phenology is a huge part of this. That overlapping of flowering is the first part of hybridization between species, and then overlapping of ecological or habitat is the second part of it. On the next slide, you can get a sense of what our overall sampling has been for this project. Each of these red dots is a place where we've collected genomic data and trait data from species. And this overlaps very strongly, of course, with the Quercus Quest campaign. You can see the oaks on the left side here. This is the list of oaks with the number of individuals that we collected genomic data from. These are also the species we're targeting in Quercus Quest. On the next page, you can see what are the traits that we're gathering for each tree. So we'll have genomic data for 700 trees. We'll have leaf morphological and spectral data. This is a different way of collecting data from trees. We have basic tree traits like diameter at breast height, estimated height of the trees, and then community attributes, what species are growing with the trees, as well as the symbionts, so that little by little we can understand how gene flow is affecting all these communities. And then at each site, we have environmental attributes of those species, of those sites. On the next slide, then, what our aim is to understand how species potential to cross-pollinate and how species ecology is shaped by and also shapes gene flow. And we know that it's very important from some preliminary data that we have in the live oaks. On the next slide, you can see that flowering time between Quercus virginiana, this iconic live oak of southeastern US, and Quercus geminata, which it grows in sympatry with it, that flowering time is displaced enough that these individual species usually don't hybridize, but their opportunity to flower in synchrony more as you go north may drive their differential rates of hybridization from south to north. So in addition to these range-wide data, next slide, your data will be used in combination with reciprocal transplant bur oak experiments that we're planting in Minnesota. I think as you click through the slide, this will give some animation here. Yep, in Minnesota in the north, in Illinois in the central part of the range, and in Oklahoma at the southern edge of the range. We're doing reciprocal transplant experiments across all three of these sites where we're also gathering phenology data. Your range-wide data will be used in comparison with these experimental plantings to understand how oaks are responding to climate gradients phenologically. So let's take a look at the next slide, which shows you an example of one of these gardens. This is the garden planted at the Morton Arboretum. This is the first 600 trees that were planted in 2021. In the lower left-hand corner, you can see that there's a really dramatic difference between these sites in the amount of leaf lobing. There's also differences in spectral reflectance of some data of some traits that are associated, for instance, with photosynthetic rate and water use efficiency. But in addition to that, there's distinct phenological differences between the populations that we planted into these sites. Let's look at the next slide. Each of these three gardens, the one planted in Oklahoma, that's the top box, the one planted in Illinois, that's the middle box, and the one planted in Minnesota, that's the bottom box. The phenology, the day of bud burst, differs between those sites, but it differs in predictable ways. Trees from trees from Oklahoma tend to break bud earliest, not surprisingly. Trees from Minnesota tend to break bud at an intermediate date, and Illinois to break bud last, except in Minnesota, where these dates change. And we have these data both for bud burst and in the next slide for leaf fall. We'll be able to relate how much of the range-wide variation that we see is attributable to genetics by comparing the range-wide data with these data from the gardens, which we expect to begin doing in the coming year. where we've got a good body of data. So in the end, next slide, these data from the genomes, these data from the common gardens, and your phenology data will really give us an answer as to how oaks adapt to climate. If you have questions about any of this, this is my email is here as well as my Blue Sky address. You can reach me either of these ways. And thank you very much. I'll pass it back to Erin.
Thank you so much, Andrew. It's such an interesting project and so many different things going on, different aspects of that project. It's just really cool. So I'm going to just take a look. I know there have been a couple questions that have come in. I do also want to make sure that I put the link for Quercus Quest in the chat, just in case anyone would like to look at that campaign too. Let me do that. And I know we've had a couple questions come in both the Q&A and in the chat as well. I might go first to, let's see, there's a question maybe for Nick that we can look at. I think this is specific to the hemlock woolly adelgid. Does temperature have any impact with egg laying? And is this also related? Has there been any negative impact from introducing the two predator insects into the eastern states? So two different questions for you there.
Yeah, so egg laying is directly associated with temperature. So as you know, with increased temperature, you're going to see egg laying happening more quickly. And that's probably led to like the relationship between temperature and insect development probably led to a lot of the misunderstanding with estivation break, where even though estivation break is happening at the same time, people thought it was really variable across the landscape because when that estivation break was happening, they were developing faster in the south versus in the north. So people were just seeing that faster development when in reality, they were likely just breaking estivation at the same time. And then temperature was taking over after that. So yeah, a lot of variability with temperature. And with non-target impacts, these are really minimal. Not a lot has been seen with this because they're specialists. They only eat hemlock woolly adelgid, so they're really just targeting that pest species. So yeah, pretty safe.
And then this is a Quercus Quest related question, although it does apply to a lot of our other plant species as well. Christine asks, for Quercus Quest, can you give some tips for making observations of tall trees? Recommendations on binoculars versus scope? I don't know, Andrew, if you want to address that. I know we've had a lot of people ask about this over the years, so I can definitely
That's a
speak to it as well.
great question. I would say from my own experience, yes, binoculars are great help. Also, strategically choosing a tree that you can reach a little bit more easily. That's a challenge because we do want observations in forest trees as well as savanna trees. With bur oaks, some of the other white oaks are pretty easy to see. Though when you're looking for the catkins you really can observe pretty readily with binoculars, and you want to look around the tree. We've even done gall wasp surveys by binocular where you're looking for just individual galls, and it's remarkable with a 2-minute survey how many you can find. Then when we subsequently go to the same trees and do some use the pole pruners to drop branches, we find that we may pick up a few more, but generally you're picking up the lion's share of the galls with the binoculars. So I think that would probably be pretty effective for phenology, but I would love to hear, Erin, more generally what you guys are finding.
Yeah, that's a great answer. Yeah, I think we definitely recommend binoculars if you can use them. Although I know some observers are observing pretty dense canopy where even if you had binoculars, you're underneath the tree, you can't really see the top. We have had some pretty creative uses of drones that we've heard of recently of people flying a drone up to the top of the tree. Maybe a special case for that. But yeah, I think binoculars is probably a good bet if you can do it. But it's also totally fine to use the question mark, and we definitely encourage that. I think some people are reluctant to say they're not sure because it makes you sound like, oh, I just don't know what I'm looking at. But the question mark can also mean I just can't see this. And we actually, because we've heard a lot about this, in our new Nature's Notebook app, so a little plug for our new app coming out next year, we're actually going to have more of a clarity on that, where you can say, I'm not sure because I can't see. And so it's just a little bit more straightforward. So yeah, don't worry about it if you have to use that question mark. And like Andrew said, we also have people that will select trees because they are a little bit easier to observe. They have lower branches that you can actually get up close to, and that's totally fine as well to select trees that way. I think we have another. There's a question in the Q&A. How are spectral data collected? I'm not sure if I understand that question. Do you know how to address that? Andrew, is that for
I
you?
do. I'm
Okay.
sorry. So the spectral reflectance data, um, what this is it's a highly specialized kind of camera, basically, that gathers light in individual spectra across a really wide range. And so they can discriminate like, you know, 2000 or something different spectral bands. Super sort of nerdy, cool thing is that when you look at different ratios of these spectra, they can correlate really well with attributes of water use efficiency, with photosynthetic activity, with oh, I don't even know what else they look at. There's been some spectral uh, ratios that have been correlated with SLAs, specific leaf area. So they're a way of gathering lots and lots of traits from from leaves in a short amount of time. We're using those in the garden as well as traditional traits like growth rate and phenology, and looking at all these in combination as ways of seeing how differentiated are populations. Because our foundational question is looking at all the genetic variation within oaks, understanding how that ranges across species, and it's huge from Minnesota all the way down to the Gulf of, from Canada all the way down to the Gulf of Mexico in bur oak, across species that have evolved over the course of the last 56 million years, and moving between species. These traits move as suites of genes that are carried by hybridization. It's probably more than you were asking, but that's
That's great.
what we're
Thank you. Let's see, we got another HWA question. Someone who's a master gardener volunteer in Maine who's been watching HWA advance. How long have the predator species been used, and has the effectiveness been measured?
Yeah, so the beetle, Laricobius nigrinus, has been released on the East Coast since the early 2000s, and then the two fly species releases started around 2015. The beetle, there is quite a bit of efficacy with that. But they are synchronized with that overwintering generation, so they only feed on that. They can prey and eat upwards of 90% of those, but because Hemlock woolly adelgid is asexual, they're parthenogenic, they're all female, and there's this density-dependent feedback that happens. This is a really kind of complicated question to answer. You actually see those populations rebound sometimes in a single year with just the beetle, and that's why we need the fly. A lot of my research actually happens on the West Coast. I do a lot of community ecology research on this suite of predators, and the fly is the one that we're hoping is going to prey mostly on that second generation. So with the combination of both of those, we're hoping to see good efficacy and control of this. And models are predicting that with this suite of predators, we actually should see pretty effective control. But that being said, just the beetle alone, we actually do see this kind of slight temporary improvement in tree health. But for long-term sustainable management, we really need that suite of predators.
Thank you. Looks like a question for Kim about, I think this is about kind of the preference of milkweed species affecting monarch breeding in Arizona.
Yeah, so what you're talking about is when monarchs are looking, I think the question, monarchs are looking to lay eggs, they ha can have preference for some species of milkweeds over others. And in Arizona, we have close to 30 species, so it's a pretty significant diversity that they can use. That is research that we are doing in the lab and more at the garden. With this campaign, it's a little more difficult because the monarch data and the milkweed data are actually kind of separate. And so we don't always know what species somebody's looking at when they're collecting the caterpillar data. Um, So we're not able to get that from this particular campaign. It wasn't one of the major questions we were focused on.
Yeah, another plug for the new app. We should be able to have that species interaction in the new app, so we're really excited about that. A couple more questions about oaks. So one was about, were there any massed crops of acorns in the Northeast, even with the drought and climate change? And that's something that you could potentially see in Nature's Notebook, although we haven't specifically looked at that yet, because you can answer those intensity questions. But you answer them in bins, so we could potentially look at that, and we are planning to do that in the next few months, actually. But yeah, Andrew, I don't know if you've specifically noticed any masting recently.
Yeah, we've observed masting. We haven't been documenting it per se. We did see a good mast in our region, the upper Midwest. In 2023, there was a good white oak and bur oak mast. We also had a good mast year in 2019. I believe it was 2019 when we were collecting acorns for the bur oak experiment that I showed you, and it was a great bur oak mast year. So yeah, we do still see mast years even as climate changes. Do we expect to continue seeing them? Masting is determined in our area in part by spring temperatures, and then when you have high spring temperatures, that can drive synchrony in flowering. So I would expect that we'll still see that synchrony in flowering happening episodically. However, masting can also be vetoed by summer droughts. And summer droughts in our area, we're seeing, and also vetoed by spring, really wet periods in the spring. So we're seeing really wet springs. We're also seeing increased summer droughts. So I don't know if anyone's looked at the incidence of mast, but it could be that it would decrease as both of those are playing at the same time, which is also contributing to oak decline in our area. On a happy note. Sorry.
And then an interesting question in the Q&A too, and I think this is kind of thinking about some of the studies that have been done in other parts of the world, looking at the mismatches between oaks, insects, and bird phenology. And I'm thinking specifically about the systems in Europe that have been pretty well studied. Have you seen evidence of that? Are you aware of that at all
I'm
here?
I, I'm not aware of that here. Yeah, that's not to say it isn't going on. That's just a literature I don't know, and I haven't made observations on that either.
Yeah, I don't know if anyone else, any of the other panelists have heard of anything. I don't specifically know of any studies in the US that have looked at that. Certainly studies looking at changes in birds' ability to track the green wave or the green up of plants, but not specifically that linkage between the oak leaves and the insects and the birds here.
There are a lot of really good a lot of good research studies cited in Teresa Crimmins' new book on phenology.
Yeah,
On
thanks
this,
for that
on this
plug.
a book, and if people haven't read this, get Teresa Crimmins' new book. It's awesome. It's just packed full of case studies. You're just flipping through it. You can't believe all the stuff that she covered in there. Super readable. And she does talk about these bird insect plant mismatches in there and places where those mismatches aren't occurring. So talking about like the boundaries of our understanding there.
I'm going to take that cue to go to the Phenology Week page here and plug our upcoming webinars. So yes, if you would like to hear more about Teresa Crimmins' new book, Phenology, just out a couple weeks ago, she's going to be talking about some of her favorite stories and anecdotes tomorrow in our Wednesday webinar. You can register for that. Let me put this link in the chat too before I forget for Phenology Week. And we also have a webinar on Thursday that's going to be digging into our visualization tool and showing you how to make your own phenology calendar. So kind of like what Kim was showing earlier with the caterpillar data, you can make something just like that, either with your own data or with some other data that are in the National Phenology Database. And then Friday, we have another researcher Q&A that will be focused on 3 researchers that use Nature's Notebook data in the last year, and there'll be time to ask them questions about that as well. I also wanted to put in one last plug, and we just announced this today, but I saw that Jackie Cluft is on the call today. So we did announce today that our 2024 Pheno Champion, our local phenology program that is our outstanding example of programs, is the Mount Auburn Cemetery Citizen Scientist. So congratulations to them. And congratulations, Jackie. Thank you for leading this amazing group of observers in Massachusetts. We have more information on our website about this group and what they've been doing over the last many years. And they're amazing group of observers. So congratulations on that. I think that we have answered almost all the questions and we're just about out of time. So thank you so much, especially to our researcher panel for presenting about the research that you're doing related to our Nature's Notebook campaigns. I think it's really valuable for our observers to be able to hear from you all about how the data are being used and kind of the importance of these campaigns that they're able to be a part of. I hope that you'll join us for the rest of Phenology Week, and please feel free to keep joining us on social media and sharing your stories and photos and all that about phenology as well. But thank you all for being here and hope to see you tomorrow. Bye, everybody.